we kindly ask you to write your questions down in the chat so that the speaker can answer them in the corresponding section of the talk. We kindly ask guys here to put your mobile devices on silent for the duration of the conference. This is Dr. Laila P. She is Director of Synthesis Lab Irapuato and also lead the research group Microbial Interactions, biochemical engineer by training at TED Monterrey, Mexico, at the University of Birmingham, UK, PhD studies at the Leibniz Institute and the Philip Schiller University, thank you, in Jena, Germany. Discovered microbial ecology and symbiosis as her favorite topics. Research focuses on deciphering the molecular mechanisms and the ecological and evolutionary implications of plant microbe interactions in arid environments and fungal bacterial viral symbiosis. Keen on developing biotech solutions based on fundamental knowledge and co-founder of Culturavi SA of CV, a biotech company. Please visit her with a big applause.
All the organizers here, especially Diego, who invited me personally, and of course all the other organizers that are making this extremely possible. I'm very happy to be here, and I like to see a lot of the young people with a lot of ideas and a lot of ideas to do. Everything is possible, no? So the only thing that we have, is the physics is not a limitation, so it's just our imagination. I'm very happy to be here. Do you hear me well? Yeah. Okay. So we'll talk today about the microbiome. And you know, in the last times, do you know how, since when we are very conscious about the importance of the microbiome? Do you know when we have started talking about the human microbiome and so on? Any idea?
Probably since the development of antibiotics.
Of course, since the Second World War, antibiotics were very important, the microbes, as producers of antibiotics and of course for curing diseases and infections and so on. But the microbiome, as part of all cells, so every one of you have more microbial cells than you have human cells, right? So we are really all colonized by microbes. And of course, we didn't see this, the importance of the microbiome before because we didn't know and we didn't have so many novel techniques to study that, right? So you know all the petri dishes, and this one culturing microbes. But now we have molecular tools that will be able to address microbiome in a broader spectrum. So I want to start, so, and of course, the Human Microbiome Project actually revealed that we are having not only human cells, but we are colonized over by microbes. So this is something very important that is changing inclusively the way that we see biology. No? Biology was like separating all the microbes, bacteria and fungi and viruses, and they are all separated like entities. But now we know they are all interface, and it's very important to know that when they are together, they have emergent properties. So they will act differently if you take it separately. So this is the main goal of this talk, to address and to highlight the importance of the microbe for the ecosystem and of course for plants, etc. But before that, probably not all of you know where I'm based. And Civil Studies is one of the leading institutions in Mexico in research, who started operations in '61 in Mexico City. But in '81, we started also operations in Silvestre, in Irapuato, in the Bajío region, because of the importance of agriculture for human life and for the ecosystem functioning, right? The plants are one of the major. So this is why we started when it was based here. And you probably will see, if you have not been to Guanajuato and Irapuato, you will see that there's a lot of colonial architecture and so, and we're here in the middle, very close to Querétaro, so you can come. And we have now two different units, the Unidad de Irapuato and the Genómica Avanzada, when we really have also all the equipment to do sequencing and RNA-seq experiments and so on, and we are a cluster, and we have two different postgraduate studies that is also very important for, hopefully for you, the plant biotech and integrative biology. And in these programs, we really develop our students, those master and PhD students, really to do high-top research. So by this slide, I'm going the other way. This is the faculty right now of the aquatic unit, and of course we have focus on different aspects of plant microbe interactions, also development, ecology, evolution, genomics, metabolomics, etc. So I hope you can search for us. And we're also increasing the number of women. So now we're around 36% in the faculty, is one of the with more women and also in leading positions as you will, well, I'm now the director as well. So I'm going to start with the topic that really is really interesting since a long time. I was living in Monterrey, no, in Monterrey, when I studied, and I realized of the impact of the sun, no, on these violence in plants and in human life. And if you see how well the planet, we see the planet, we see that at least 48% of the world is really covered by what we call dry lands. And how we define dry lands? Dry lands is we can have even like an index. Aridity index is what defines the dry lands. And you can see, of course, the broad is the hyper-arid regions, and until yellow, the dry and arid regions. And this is meaning just the precipitations of how much rain do we have, and how much of this rain is evapotranspired during the potential for losing this rain again, right? So this is the aridity, and as you can see, well, this is very, well, a lot of the earth is covered by these islands, and of course Mexico has more than 50% of the territory in this region. We are in one of them, this one is also, and of course Irapuato as well. And something that is very important for us scientists to combat as well, and to bring out new solutions, is actually that the climate change is affecting these biomes, these island biomes, in very hard ways, like increasing temperature and of course increasing growth. And this is something that will impose a lot of challenge for life to continue as we know it. But of course, in these arid environments, also nature has already created different solutions. And you have seen, for sure, all these different plants, like they are surrounding us, always they are there. And of course, these CAM plants, they have this photosynthetic metabolism that is already very efficient. Which one? Well, do you know what is the photosynthetic metabolism? No? So anyone? Yeah.
I believe that they only open the stomach to absorb gastric change during the night when there's not a lot of sunlight, so they don't use that much water and maintain it close during the day.
Exactly. This is the most efficient way of doing photosynthesis if you are in a hot area, right? So you close your stomach during the day, and the transfer of the gases occurs in the night, and the fixation of carbon. That is the most important thing that they can do, well, many of the things that they do. And of course, so these plants, especially, they have developed this kind of metabolism. And of course, they have a lot of other adaptations, like you see the wax, the cuticles, they are very big. They have lose the flowers, but the leaves, not to lose all these areas. And of course, also in the roots that we can see here. But something that was missing is actually what role do microbes play in these plants? Are microbes associated with these plants that also help them to overcome these hostile environments? So this is my research question when I started here at CIMERSAF. And of course, cacti are very important plants. There are more than 2,000 different species, and they originated in the American continent. So you will see cacti naturally from the south of Canada to the Patagonia, and of course in Mexico, we have even like the Tenochtitlan, no? Tenochtitlan, like the big symbol how to crown Mexico Tenochtitlan, no? So the people that were coming from the north, probably through the very passage, they have to see the signs to really establish, no, Mexico, no? And of course, cactus has a lot of different uses, including probably some of you have been to the 14. You can see that you can have alkaloids and different compounds in cacti, and of course, there are even for diet, right? So we have for Ataquiza, we always have the Volatiles and so on. And they have really a huge, like I said, from a plant perspective, no? They are really very unique plants with very different, but very interesting plants as well. And something that is already now becoming even like a very big issue for Mexico, because now we are selling a lot of mezcal and tequila all over the world, especially to the US, no? It's the first consumer after Mexico of tequila and mezcal. And you know, agaves are the main plants that are used for the production of these two beverages. And of course, they represent Mexico all over the world. And they are not, these plants have been really used since prehistoric times for many different things, almost for everything, for making your house, for even for rituals, and of course now we are in the transition of using also this plant for biofuels, for probiotics and different things. And Mexico, as in the case of Patty, has also the big diversity. Indeed, the agave family originated and diversified in the Mexican country, in the Mexican territory. So this is why it's also like a responsibility that we take care of these plants and these lineages because they originated here and they are part of these ecosystems that sustain life in these islands. So based on this, of course, these plants, they are differently phylogenetically, the agaves in monocots and the cactus in dicots. They produce flowers and everything, but they are different families, plant families. Actually, which one do you think is older? Which one do you think are older in the history of?
I put that idea because you mentioned that are
Well, interestingly, the cactus is older. It's like 30 million years old. And the agaves is a little bit younger. They are supposed to be like 10 million years old. Of course, but they diversified distinctly, and this is interesting anyway that they have this difference. But something that we can ask when we see, when they most of the time they coexist, they live in sympatry, is what we call it. So the question that we ask is actually which microbes are living with these plants in these environments, right? And of course, how are these communities assembled, no? How they are assembled and how they are selected, no? Of course, if the plant exerts a selection power or the environment, or how is this selection occurring? And at the end, we want to know also what do they do for their host? Are they helping these plants to survive these environments? Can we manipulate also the microbiome to make them better or to make more resistant, etc.? So these are the questions that we have. And for this one, something that is very important in microbial ecology, as in most of the science, is actually to do a design, no? An experimental design that actually helps you to cover the topic. So we decided to study wild and cultivated agave species and cactus species. So I will show you we have some samples from California with some collaborators. And of course, in Jalisco and Guanajuato, where agave and cacti are also coexisting. And we sampled 5 different species, including agave tequilana, the one that is really just cultivated for tequila production. In Querétaro, in Querétaro, there are not so many, but in Guanajuato, it's not a denominación de origen state. So Jalisco, the whole state, and Querétaro and Guanajuato have denominación de origen, so agave tequilana is growing for the production of tequila. And we have here agave de Serpi, agave samiana, and these two cacti that we have here, Neculocactus, the Vetisans, there are plenty of these plants here, and the Puntia robusta. There are also like sympatric or wild species. And something that we consider also for this design is to see how the impact of the season, like the dryness and when you have some rain, how this affected the microbial communities. And something that most of the people sometimes think about microbes and plants is just the rhizosphere. And the rhizosphere is just the niche of the plant that is more studied. The rhizosphere is the soil attached to the roots, and that's what a lot of interchange is occurring. But now we know that plants are colonized all over, inside and outside, and also in the above ground tissues. So also what we call the xylosphere, like the equivalent to the rhizosphere, but it's just the surface of the aerial parts with the air. And of course, the endosphere, so the endophytes, the ones that are living inside, and the leaf and the roots. So we're using molecular methods to determine which communities are there and what they are doing. So in the next one, I will show you something that we, after sequencing, right, like the 16S, one of the molecular markers for bacteria and archaea, and the ITS2 for fungi, we realized that they have more than 8,000 microbial different lineages of these plants, no? In the different compartments. So you see that they have different patterns. You see by where they are living, where the microbes are living. So in the soil and outside, here bacteria here, and the phyllosphere, including like specific and fungal endophytes. But this tells us already that these plants are really interacting with many different microbial communities or different microbial communities along the years. And of course, when we do this analysis, how important for the biodiversity, so how important is all the factors that we've been measuring, like the species, the sites that we have in California and Mexico and so on. And something that this diagram is telling you is actually, you can compare, like, if the points are closer, it means that the communities are very similar. If they are separated for a long distance, they are very different. So you can see, for example, here that the soils, the bacterial communities in the soils were very similar among all of them. It doesn't matter that they were like 2,000 kilometers, like from California, Guanajuato, Jalisco. And as we move in the plant, so within the plants, we see that what is outside the plant is here, the epiphere. So the rhizosphere is in red, and in blue, the phyllosphere, so what are living in the. And then we have the endophytes in the roots and the endophytes that are living in the leaves, outside the leaves, no, in the surface. So this analysis with all these samples in these years told us that the plant compartment is very important. So different communities, bacterial communities are living in the different spaces of the plant. And how the fungal communities look like. And this is exactly with the same DNA, now using just the Volatiles 2 marker, you see that we have like the Rio Grande in the middle. You see all the communities, the fungal communities from Mexico are in the left, while the California communities from the US are in the right. No? So the fungi, these into bacteria, they have really bad geographic patterns. And they start with the soil. So the soil already, you can even use now the shoes of to see a fungal analysis will tell you where you have been, because the soils have specific markers. And as you can see, if you go inside the plant, it's different what is inside the plant and what is selected from the environment. No? As you can see, like from the soil and the physical. So it's a clear distinction what lives inside the plant, what is outside, and also this geographic patterns for the fungal communities. Yeah.
I have a question. So, how do you compare like the bacteria or the fungi?
How we analyze, we extract DNA and amplify two different markers by PCR, and then we make a libraries that you sequence. And we have for this data set, we have like millions, 20 million sequences that we can tell. So all the communities. So when we are comparing these two different points, for example here, so when they are very close, it's like comparing like the sample from the phyllosphere, from agave deserti, for example, from the has 10 taxa, more or less in these proportions, and the other one has the same taxa. So when they are very similar, they have the same more or less taxa, and also more or less in the same composition, relative abundance and so on. So that means this analysis try to simplify when you have multiple factors that you can see which ones are the most important. So you can see the patterns. This could have different patterns as well, right? But in the case of this sample, it was very useful to actually see the global patterns of distribution of both bacteria and fungi. So I don't know if I was clear enough or okay. So that's why, well, now we can consider, well, something that we are interested in as well is how the diversity is affected, no? So once we have like this census of how much bacteria, how many fungi, we can also quantify the diversity. So is the diversity important for the functioning of the ecosystems? And something that you can see here is one of the index that we use for diversity, that is the Shannon index. And as you can see here, well, probably most of you, even if you are not in biology, you have here that in 1 gram of soil, you can find millions and millions of different taxa, right? And it's something that you can see here also confirmed. So in the three species of agaves, including the cultivated one, and the soils look really very similar with very high diversity. And as we move into the plant, the diversity lowers a little bit, especially the endophytes. So not many microbes live with the plant when they are healthy enough. They are very selective. But something that was striking in this finding is actually that the fact that Agave tequilana, the species that we cultivate, has a very low diversity in comparison to the wild agaves. So that means or suggests actually that when we are manipulating our crops or agaves, the agaves are losing diversity for management. So this is very low because this is a log scale. So it's really very significantly low compared to the agaves, the wild agaves. And when we use also network analysis is very helpful to see not the things separated, but how they are acting together. And probably you will be able to see here, this is the network form of Agave tequilana from the phytosphere, this compartment. And you see here probably that you see the green dots. This is just fungi, and this is bacteria. As they were not getting along together, but separated. And in Agave thessaliana, the wild agave, you see that they are more interconnected and more robust. So this actually telling us that Agave tequilana is suffering some kind of reduction in diversity and how the microbiota is active. And this was just a suggestion based on our data set. But we wanted to go further, and now you can also use short-term metagenomics. That means that you have a DNA sample and you sequence everything. You don't do any amplification a priori, so not a PCR, but everything. And by doing this analysis also with these communities, we actually can see that some functions are actually missing or reduced in Agave tequilana now. So this is a diagram, a triangle diagram. You can see cacti, Agave tequilana, Agave thessaliana, and you can see that some points look more like present in some of the species than in others. And actually, I will just go through this very quickly, that you can have a look how these things are analyzed. So we detected that phototrophy, the capacity to use light to form carbon, to be photosynthetic, is more present in the cacti. You see these blue dots here, 68–80%, and also in Agave thessaliana, but it's not anything of this in Agave tequilana. So Agave tequilana and the microbial communities don't have these functions. So actually telling that it's not just the phylogenetic reduction, but it's also functional, right? So something that we asked, of course, like the next question was actually, despite all of this, because we are one of the challenges of using the microbiome is there are so diverse, there are so many, how we can study them, all of them, and also in interaction with a host. The same happened with the human, no? So now a lot of the studies are correlative studies, no? You see something is going high, but you don't know it's the causality or it's the effect. So this is something. So something that we are aiming is actually to have some core microbiome to see if we have some core microbes that are the most important in one ecosystem. And in this case, you can see here, despite this difference in geography and the species and so, we have really a core microbiome that is not so many of these taxa that are shared across all these environments and species, and that they are the most abundant ones also in the communities. So probably if we study this core microbiome, we will have some rules or some functions that are very prevalent in this environment that we can also manipulate. And this is one of our aims. And so now that we have described this, the first step was to really describe who was there, what were the patterns of diversity. Now we want to move to function, right? What are these microbes doing? Now that we have, we know who is there. So this is the field of Agave tequilana. You can see this is in Pénjamo, very close to Irapuato. And of course, this is one of the most important also crops now in Guanajuato. So I will talk today to three different ways we have addressed this question of functionality of the microbiome. I will talk about the volatiles, about synthetic communities and about mycorrhizal fungi. That is one of the most known symbiotes of plants. So volatiles, you know, because even every time in the morning, if you smell the coffee, you're ready to go, right? So the volatiles are chemical odors that we perceive, and then even we react to this odor, right? So even if we see, and the species interactions are also mediated by this chemical communication. And this was first discovered actually by pollinators, and so how they attract the female and the male is also because of these odors. And of course, this was even a Nobel Prize, and what we call most of these chemical communication started with plant insects interactions or between insects. But now we know that plants, of course, have will use all these defenses also for attracting pollinators, to defend for herbivores, and even to plants, parasite plants, can detect the plant that they like because of the odors, right? So this is one very special case. But something that just arose came in the beginning of this 2003 is actually that microbes, of course, they produce also all these volatiles, but they have never been studied. And as you can see here, for example, in this example with this petri dish, that different colonies, strains of bacillus, were able to promote growth of Arabidopsis, one of the model plants that we use a lot in biology, in plant science. And of course, that was the first report that bacteria also produce volatiles that can also change plants, right? Not only between plants communication or with the insects, but also microbes. And microbes, now we know they are living always in the plants, right? They are not just when they are pathogenic or so. So we started this question like, well, the core microbes that we already detected, do they produce volatiles that can affect growth? And this is our main question. So you see, we screened not so many different 40 bacterial strains and 22 fungal ones, and they are very abundant in the microbiome of agaves and haki. And the most important thing from this one is we make this screening like very synthetic, no? So you have a petri dish where you have separated the plants growing in one side, and in the other side you place the microbe that you want to screen for volatiles, but they never touch, no? There is no physical contact. The only contact is through the atmosphere, through the air that they are sharing. And as you can see from this experiment from our bacteria, one of the subjects of the screen and the fungi, you can see that the plants change a lot just by the presence of the microbes. That means that there are a lot of volatile signals in these petri dishes that the plants can sense and react. And of course, if you see very in detail, we see for example here with this fungi that they have really a lot of more roots and a lot more foliar, so they are very promising. So we continue and actually did the GCMS with gas chromatography and mass spectrometry. We detected more than 120 volatiles produced by this bacteria and fungi, and they belong to different chemical classes. And I put here the most prevalent ones. In bacteria, we have a lot of compounds that are belong to the classes of esters, and the fungi to the terpenes, that are very big chemical family that have a lot of different functions and also influence. So you see, it was very work, but how we can distinguish these single compounds from these a lot of compounds? So we did a screening, and after, so from this library of more than 120 compounds, we could buy some that were already being sold, produced in pure compounds, and we identified these 8 different compounds. As you can see here, esters, alcohols, and terpenes, and so on. And something that we detected, of course, is that some of these compounds have, well, they are dose dependent, no? So the compound is bioactive, but depending on the doses. And you can see this here. If you have a doses of 50 micromolar, you see a lot more growth compared to the control. But if you put a lot of this compound, the plant inhibits its growth, right? So this is chemical communication is a lot about not just the right method, but also the volume, no? How much of these methods is really in there. But this was very interesting already because from this screen, we have like they increase the number of roots, the main roots, the lateral roots, the foliar, the biomass, and so on. So we were keen in not using this knowledge just for Arabidopsis, that is the model plant, but actually to see if this happened in agaves. So we moved to agaves because they are more difficult and more long living and very slow to grow. But you can see that some of these compounds had exactly the same effect as they had in Arabidopsis and now in agaves. No? You see agave tequilana growing with this alcohol, 50 micromolar is really expanding the roots. And the same happens also with agave samiana and some other esters. And after this treatment with volatiles, microbial volatiles, you can see that the impact was even strong enough that after 8 months, we see still the effects of these treatments. Sorry. You see, like the effects are there, no? So this is very important. We have investigated some patterns because, of course, now we can increase the growth or promote the plant's growth even very dramatically. So with one single compound from bacterial origin, right, or fungal origin. And of course, I was already pointing to this problem that we have as Mexico, because many of the species that are used for mezcal production, they are not even domesticated, or they have not been really, they are just in natural populations, and some of the people just go and collect the piñas after 10 years or 15 years of growth. And a lot of the populations have been now are decreasing, and it's really a problem for the ecosystem. So we are interested in trying to help this also to sustain mezcal production and of course these biomes. And we have now tested these volatiles of these mezcal species. We also have very good results. So actually that's why we are starting also this company, because now we want to transfer this knowledge and to really make use of this technology for improving the capacity to restore these agave populations and of course improve the productivity, and that the people in these regions can live from this, benefit from this plants. I mean, I would say. So with this, of course, we just started. Now we are in the iceberg, the top of the iceberg, because we don't know so much still about chemical fermentation. So if we have so many steps, one of the things that we don't know is how plants actually sense all these volatiles. Why do they react to some of them, and to some of them they don't do anything, right? And why are these changing the developmental program? So by this one, something that is very nice now, with Raul, a PhD, recently like graduated last year, together we co-direct this thesis. He designed a new GCs instrument to measure volatiles on time, in real time. No? Now you can put like this equipment that you will see probably later, and we can now measure the volatiles emissions. Right now it's a batch process most of the times, and some of them all the TCMS are very expensive, and most of the universities cannot afford to have one. So this is something that is limiting actually the field. So this MOBIS is also very good for that. I will show you some data. Of course, now that we have genomes of all these modern microbes, for many of them, we can also investigate how these microbes are producing the volatiles. If they are producing in a regulated manner, in reaction to the plant. So when are these volatiles produced? So we can now study the biosynthesis also. And of course, we are studying why the plants respond to these volatiles like this, no? What are the programs, and what happens if you receive signal 1 and then signal 2? No? Do they synergize or? Yes. So this is something that we will do. So this is equipment that Raul developed in Semesta, and this is very small as a portable equipment. But now we can measure in milliseconds actually the volatiles that are being emitted by the plants or by the microbes. And as you can see here, for example, we can track the dynamics. So you put one of the volatiles in this chamber that is with flux, and you see cinetic emitters come out. And when you have the plant absorbs some of the volatile, and the time of the peak reduced. So this difference is like what the volatile was absorbing the plant, right? And later, with some other techniques like plasma and imaging of the tissues, we can detect, you can see exposed leaves and not exposed different tissues. And we can see that the volatile, this volatile that was just appears to be in the stem, no? In la piña, from the agaves. You see here the sign, the mass from this compound, and you don't see that in the not exposed plants, right? So we can now detect where do the volatiles go and what do they do in the plants. So it's very promising, and technology is allowing us to answer this kind of question. And now I will move to the synthetic communities. I think now that we have this ecological framework, that we know who are there, something that we can ask is, well, what we assume is that we can design a community that represents in some way the community, the whole community, no? To some scale, no? So this is, we cannot cultivate the outer 8,000 microbes. We have just some in the lab, but we can tell, for example, in this one, we are using the connections that we have seen in nature to see who are the leaders of the community, no? So if we have the leaders, they will probably call their friends, and they will recruit their friends, and then the community will establish, even if we just have, like in this community, we have 5 members, no? Just 5 members, you see Methylobacterium, micrococcus, different species from bacteria. Here we have the fungi, here we have bacteria and fungi. And if you see these nodes, they are the leaders that we think are the leaders, are here in green, for example, in yellow, and we will call all the other ones, because this is what the ecological data set follows. And the other thing that we designed synthetically was based on the functions that we observed that were enriched. For example, cyanobacteria makes photosynthesis. So if we include photosynthesis that agave tequilana didn't have, now what happens to agave tequilana, no? Of the communities. And of course, we tested also the random communities. If we don't have any framework, ecological framework, if we just choose different microbes, what will happen then to the community, to the communities and to the plants? And this is I'm very happy from this experiment, because of course it was not easy to have all the leaders, no? And then the first thing that you have to do is to culture them, right? To try to make different strategies to culture the microbes that we don't have in the collection. And in this case, more than 200 microbes were cultivated and isolated and characterized, that you can choose which ones are the best for using in the synthetic communities. And then now that we have collaborations with the tequila producers, we have in Pénjamo, there's a lot of fields, no? So they are really homogeneous, and you can do your experiment there based on these collaborations. So we have 1 hectare of agave tequila plants already growing in the field, where we can ask these synthetic communities and check what happened then to the plants and to the communities after the time, right? This is what we have done. So these are plants that were just recently placed in this field, and then we have the zinc added, no? With this design. And something that we realized after sampling all this time is that it was very important for the phylosphere, for the external part of the communities, is actually that we can see, you can see here like in the same data set as I showed you at the very beginning, but this one is just from this community. And you see that the season had a very strong contribution on these communities. Not so much the synthetic community, this here or there, but more the season. And something that is striking is we have less diversity when it's rainy season. You see here, the lower diversity in richness and so on. And the same in the fungal communities. The fungal communities are really like telling exactly which time of the year you are, no? Not only where you are, but also the time of the year. So the rainy period is all together, the dry season, depending on that. So this was important, but of course, something that we wanted to see is actually how diversity was, if this changed or did not change. And as you can see here, bacterial diversity increased with these communities, especially with the one that was inspired in the ones from the wild agave, as you can see here. And also when you see the network. So not only the diversity, also the metabolic diversity, the capacity to use different carbon sources, as you can see here, for example, in comparison to the mock plants, no? You see that's a very huge increase. And also how the communities now are integrated. And you see that the networks are much more integrated than here, no? And this looks very similar to the previous results that we had years before in our sampling. So this was telling us Yeah, we have manipulated the microbiome, and now the communities are more diverse. But do these communities affect the plant? This is the most important thing for the producers, right? They don't care so much about diversity, they care about at the end, if the product will be better or have more sugars and so on. And this is something that we also see. So after 2 years of this experiment in the field, we see that the amount of sugars that the plants are accumulating, this is the most important thing for agaves, because from these sugars is what you make even biofuels, or you make also the tequila or the mezcal. So they have a bricks, these private bricks that you measure, and you can see in the second year, the scientific communities really make even like one year ahead of the other plants, right? So this means like the productivity of the plant was increased as well, just by manipulating the microbial communities with these symptoms. And this was just published that last December. Is one of the editors picked this paper from us in Partida-Martínez. So Victor was just graduated. He's now doing a postdoc stay in Germany in mathematics too. So he's doing well. One of my students. I'm happy to see it. And the last step, just to finish. Probably you know that arbuscular mycorrhizal fungi is also very important for plants, right? They are the oldest symbiosis known. They are even in the fossil record, no? And of course, for agaves and cacti, they have also been showing positive effects, no? They are like mutualist symbionts of the plants. They produce, like, they can give plants phosphorus, water, and so on. And now what we are investigating is also the bacterial component in the arbuscular mycorrhizal fungi. So you see these mycorrhizal, they are biotrophs, so they need the plant to reproduce. So you cannot have a petri dish which a mycorrhizal fungi. You need the plant for reproducing fungi. But we have detected now that all these fungi have also bacteria living in us. And these bacteria also play some roles in this tripartite symbiosis. You have the plant, then you have the fungi, and the fungi have bacteria inside that can do different things for the symbiosis to occur. And this is something that we just started to go in deep. You can see here this Pac-Man, so it's one of these spores. And of course, with some microscopic techniques and staining, and so we can see that bacteria are inside. And we have at least two different groups. One are the Volatiles, and the other ones are Volatiles. And of course, they were investigating now what do they do for this symbiosis. And something that just a preliminary experiment that Daniel did is actually how is this symbiosis affected by drought? Now we know that in the future, we will probably have more drought in this in general. And of course, these plants, we always think that they can resist drought because of the harm and all these things, but we actually don't know what will happen if the things continue like this. And you can see here in this experiment, we manipulated the amount of water that the soil had. So for mass 70% boil water, relative humidity in soil, until 20%, that is really very dry. And for 8 months, right? The normal 8 months that the plants are inside. In this case, the data set that I present here is after 6 months. And you can see, well, first, this is the plant, the agave tequilana plant, without any mycorrhization in the middle row treatment. So you don't see any mycorrhiza forming. And of course, the plant is not very happy. The plant here is the same, the same genotype, but of course, here is with the fungi being colonized. You see here the vesicles and the leaf. And what we have seen, of course, is very interesting, but that of course, many of the parameters of the plants are much better, right? Like biomass, chlorophyll, and so on. So actually, the fungi is really helping. But something that I'm not presenting here, we saw that in the severe drought, the symbiosis is prevented. No? There are no more colonization. So we think now that drought is a very important modulator of this symbiotic interaction. And if the drought is really severe, we will not have these interactions, even if they are positive. No? So we are actually. And well, in summary, hopefully, I came with the ecological framework of what we have done first to investigate which are these communities. But of course, once we have this basic knowledge, we could move forward to the different topics that I showed you, like the volatiles, that now we have more than 8 different bioactive volatiles that we can test how they work in plants and even use for increasing crop production, as you can see here. And of course, these syncoms were one of the others also designs for improving, changing microbial diversity and improving plant productivity. And as I showed you here as well, now we know that mycorrhizal fungi and the bacteria that is living inside mycorrhizal fungi may play a role in growth tolerance also in agave tequila. No? How we are continuing, of course, there are more questions and answers, right? And I don't know if you have any questions. And of course, I would like to say this is my my research group at Simba. And of course, we have had a lot of support from CONACYT, from the GDI in California, from the secondary in Germany, the Oak Ridge National Laboratory in the US, and of course from the tequila company that have supported and helped us to do experiments and so on. And I thank you for your attention, and hopefully you have some questions, and I will be happy to see. And this is the last harvest of PhD students that I have in my lab. All these 4, the ones that I presented in the last, we had a really difficult time, especially because we have all these 2 years, almost that they didn't have really like an open lab and support to the technicians were not going to see the lab and so on. So we did all this work together, and it's Raul, all the just graduated, Danny is the last one who hopefully will graduate in the next month or so, and they just really want to make it. I'm happy to take any questions. Thank you.
For me, it's very good. I was wondering how affected are these microbial communities by global warming?
Now, this is something that is very hard to know exactly right now, because we have exactly we have to do yearly assessments, right? And the microbiome is changing also along the development of plants, no? Exactly as it changes with us, no? Like we have like, for example, for humans, we need at least 3 years, every 3 years, to the microbiome to settle down, and then to start like on the process, no? Different ways. So with these plants that they take 7 or 10 years to develop, we have not made all these analysis throughout the year, right? So right now we are going also like exploring also the seed microbiome, because in the case of agaves, some of these they produce a lot of seeds, and of course there are some vertical low-grade transmission, but they are recruiting the microbiome along life, right? And in the other things that we have done, we can make probably 2 years sampling, and we can check what happened in the first year to the second year. But we have not now an overall view how it is, especially now that it's very marked that the temperature is increasing and the growth is increasing, so we don't know yet. But of course, it's something that we would like to address and to have more funding, like we can really do this in a long term, like 10 years, 11 years, 20 years, but we really monitor how the microbial communities are reacting to these changes. And of course, what we have learned from this is actually, it's not only important, is the plant communities are important. If you have different plants in an ecosystem, every plant has their own endophytes. It's something that I didn't highlight so much, probably because there's no time for anything. But of course, plant diversity correlates also with microbial diversity. If you start to decrease, for example, if you eliminate agaves or eliminate the other plants, the herbaceous plants, or in an ecosystem, you are losing a lot of microbial diversity too, and that's why we will have problems if we don't know how to manage these ecosystems, but more from the ecosystem point. But the microbes must be always investigated, because they are the pillars of this ecosystem as well. So, but we don't know yet. We went through, and I was very amazed by this that the drug modulates the symbiosis, right? So if it is extreme, they will not do symbiosis, no? Because, so it's very, yeah, so that's why we have to be very careful. And of course, a lot of, I think something that in the microbial ecology, well, most of the people don't realize of the importance of microbes because they are not visible. So something that the community is trying to highlight how we can make more visible these little tiny things that are really important, and of course that are linked to animal diversity, biodiversity. So it's important, but yeah, hopefully not so much. So that we can do something with it. Yeah.
Yeah, so I'm absolutely in awe with the research. It's wonderful. Congratulations.
Thank you very much.
I was wondering about the effects of domestication, because you mentioned that the agave tequiliana, the more domesticated species, kind of had a very different and less varied microbiome than those wild.
One relatives, yeah.
So I was wondering if something, for example, in Arabidopsis, which is a plant that we use to model basically everything in plants because it's really easy to reproduce.
Yeah, the
whatnot.
collection of milk and everything else.
Exactly, but perhaps in our tinkering with the plant, do you think that maybe we altered the microbiome of the model plant, and perhaps could that be extrapolated to other domesticated plants that we may use in agriculture?
Yeah, of course.
The yield might not be as high because of this?
I think it's something that we will do. Like one of my aims is actually to transfer not only this to agaves, but also to tomato or sorghum or maize or so on. So it's something that we need to do. And of course, what we learn from this will be useful for agriculture. In our agriculture that is already synthetic, right? And I think one of the values that we have to keep is to keep the natural environment as more pristine as we can have. But of course, investigate them. They are the resource for everything else. So what we have learned from this and many other people, not only us, not even in maize or in beans and so, is that the wild relatives that are living in the natural ecosystem have a lot of more tools to defend themselves and this is the things that we can transfer to the way the cultures that we have, even with new ideas, right? Because, for example, I imagine also symbiotic systems when I can manipulate completely. And then this will be, but they have to be in a contained space or area, right? So we don't want everything to be equalized at the end, no? Because we will lose the diversity. We just, so we have to keep this difference. For me now, because now we are doing some experiments in Oaxaca, Puebla, where there's a lot of diversity, and I think it's very clear for me as well that the people need tools that they really can handle, right? So you cannot just tell them, do this, it's very good, but if it's very, very tedious, they will not do it, and it will not be visible. So we have to keep these things separated, in my view, but study a lot of the nature as it is. It has all the lessons, and then with our creativity, we can do the things that we need. But they will be separated, they will not be like, yeah, this is my point. And of course, it's very useful always to have the wild relatives, and domestication is having a lot of impact, and because of many different aspects. And you see, for example, in tequilana, because the plants are not allowed to reproduce, right? So they hold this big skin because they have all the sugars, and then they come the floral bouquet, and so the plants are not there to reproduce. And that's why we have also a genetic production in the genome of agave tequilana. And of course, that's linked also to the microbial diversity. No? If the genome is already reduced, the whole genome is also affecting the microbial diversity, plus or cultural practices as well.
We'll have to stop the questions if we need to move on to the next one.
Okay, yeah.
But you're gonna still be around.
Yeah, you can, of course, I would be happy to talk to them and write if not the case. Okay. Thank you very much. Thank you so much for everyone. Sí, eso, los volátiles son más bien como que cambian el desarrollo, como si fueran hormonas. No sabemos con detalles. Sí, claro.
Buenos días, doctora Robles. Te voy a poner como en un momento como co-host para que puedas compartir. Ya debes de poder compartir la presentación.
A ver, denme un minuto.